And could you please tell me that you can see my screen? Just confirm that you see my screen.
Yes.
Okay, thank you. Okay. So, as I mentioned in the previous session, we kind of wrapped up most of the biotechniques that we utilize for DNA analysis. And this week, we are shifting to RNA analysis. We will have 2 sessions. And after that, we are going to move to protein analysis, which will take us maybe 4 sessions. And the last 2 sessions will be broad analysis, echocardiography, and flow cytometry. So in this lecture and in the next lecture, we will discuss how do we use techniques in studying the RNA. Particularly, the most interesting and the most widely used is the gene expression profiling. Probably within this lecture, I have to go very slowly on the concepts so that you know the gene expression profiling and the quantitative real-time PCR or qPCR. Once you know these 2 steps, then we can decide to discuss more on the microarrays and the RNA sequencing and the single cell analysis, because these are quite similar in bulk, while quantitative PCR is done for a single specific gene. Now, you all know that RNA play many important roles in the cells. One of it is that it's the mediator between DNA and protein, so it's playing a role in the protein synthesis, and therefore expression of certain genes. So it can be regulated by RNA. It can be transmitted to proteins by the translation process. So module 11 is important for you to recall most of these processes. Now, as I said, these 2 lectures will be discussing how we isolate RNA again. I will refresh your memory again, and then we will start describing the qPCR. So most of our discussions will be from the step of transcription to the end of the translation, to the beginning of the translation, sorry. So an important aspect that you have to keep in mind when you are studying RNA is always the process of splicing. We already discussed replication and transcription before many times. I want to focus that you have to be very careful when you are studying a specific gene by looking to how many transcripts it has, because the splice variants mean different messenger RNAs in case they are coding or not. And that can affect a lot the results you're looking at. So from the process of transcription to generate, in this condition here, a specific type of RNA, which is the messenger RNA that is translated to a protein, these steps can offer multiple levels of control for the cells. For example, the cell can turn on the transcription at certain point, or it can turn it off. Or in other words, it can also be affected by certain messengers that come to induce the transcription. Like, imagine there is a cytokine that is needed in order to bind to a transcription factor that turns on the control. So this we have discussed in the previous slides. I don't want to go back to the deep details, but it's important to know that while we focus on messenger RNA most of the time, because that is coding for proteins, we have many other types of RNA that we will need to discuss. So in terms of the RNA splicing, remember that different exons can lead to generation of different transcripts. So in this particular example here, we had 3 exons. The first isoform has exon 1 and 3. And the second isoform, it has the exon 1 and a truncation of exon 2 and exon 3. So in practice, the DNA, the genomic DNA is the same. However, the messenger RNA from these two are different. And today we will today we will discuss how we can identify this isoform from that isoform by qPCR. Okay? So this is very important to take into consideration. So one thing you have to always keep in mind that we discussed many, many times that RNA from the transcription, it passes by multiple levels of checkup, and there are parts of that RNA that are not translated, and there are caps that are added on the 5' and the 3' for the stability, and then the RNA will become stable and in the final version, which is called like the mature RNA, mature messenger RNA. So for today, we will start from 0 in terms of the isolation of RNA. Just to refresh your memory, that imagine today you will do an experiment to describe how the transcripts in one cell type is different from the other cell type. So simply you have cells in culture or tissues like heart or liver or muscle, and you want to extract the RNA from that. And after that, we will start describing how we do the primers for a qPCR, and then we start analyzing the qPCR. So recall that when you are isolating the DNA or the RNA, you have to be careful what is the starting material. And you have to be careful that whenever you do the disruption of the cells, then all the components will be mixed together. So the RNA, the DNA, and the protein, they will be mixed together with the membranes and all the compartments. So the steps that come next are important to know which molecule you are isolating. So for example, you are isolating today the RNA. So you have to put the strategy in order to get the best type of the RNA. And this strategy would be after lysis, whatever process of lysing you are using, detergent or mechanical. We discussed the types of lysis before. Then, since the RNA is very sensitive, you have to directly protect it by killing the RNases, by using RNase inhibitors, so that any RNases that break RNA is inhibited. You also want to get rid of any proteins, and particularly you will do a digestion with the proteinase K. That's in many kits that we use in the labs. You will find this component, and you will digest any remnant DNA or genomic DNA, because you don't want to have a contamination of your RNA with the DNA. So many times we do DNase digestion for the sample, so that in this step, you are assuring that you will not have any more proteins or DNA in your samples. Remember, if you are using the RNA extraction method with the phenol, it should be the acidic phenol, which is a lower pH with guanidinium salt. And then you will do the recovery of the RNA. You will wash it to get a better purity of that, so you don't have any salts. And then you will resuspend it with water or whatever buffer that you have, like TE. And finally, you will have to be very careful with these salts that are used. Some of them are volatile and toxic for your health. And if you perform such type of isolation, obviously you are isolating all the types of the RNA, not only one type. And in this screen here, you have transfer RNA, messenger RNA, ribosomal RNA, small nuclear RNA. So there are many types of RNAs that are isolated. Previously, I discussed with you a couple of those RNA types, sorry. For instance, we discussed the messenger RNA that is generated by the gene transcription. And we mentioned that it is the one used for the COVID-19 vaccine. We discussed about the transfer RNA and the ribosomal RNA when we went through the transcription and the translation process. And we described the microRNA and the small interfering RNA when we want to knock down a gene. So we want to kill certain RNAs that are specific for a gene so that the gene expression doesn't continue. And that's how we achieve gene silencing. So if you have questions or you didn't go through module 11, I strongly recommend that you do that. Now, if we want to describe the RNAs in terms of their functional aspects, we can divide them into two different categories. The first category is the coding RNA, or the cRNA, and then the non-coding RNA, which is an ncRNA. Now, the coding RNA, these are RNA that will be translated into proteins, like the messenger RNA, the transfer RNA, and the ribosomal RNA. So these are the category that is translated into actually a protein that plays a role as a protein compartment, a molecule, a biomolecule. However, there is a lot of the genes that are encoded into RNA, but then they are not translated to protein. And this is the non-coding RNA molecules. And some of those are housekeeping. So the non-coding are the transfer and the ribosomal RNA. And the regulatory part, which is many of these, we don't know what is the function. Like there's tons of long non-coding RNA that we don't know what is the function, but in the last 3-4 years, the long non-coding RNA is emerged as one of the categories of the RNA that is very widely studied, and it plays a role in the gene transcription and translation. So it regulates many genes. The microRNAs and the small interfering RNAs, we already discussed those. I hope I will have some time to give you an example of the long non-coding RNA in the next few sessions. Now, just to refresh your memory, if you forgot how the microRNA and the small interfering RNA work, these are RNAs that have some inverted repeats in their sequence. And those inverted sequences cause the formation of these loops. And once these loops are generated, so in condition of the microRNA, there will be a transcription process, and that transcription process will generate a messenger-like RNA that it has these folding sequences or palindromes or inverted repeats, and therefore you will generate these loops. Now, once these loops are generated, that's where the RNA interacts with the protein called dicer. And then the dicer will cut those into smaller 21 to 23 base pairs. And then another protein called RISC, it will bind, this protein is gonna bind with those 21 to 23 base pairs. It's gonna use only one of those as a guide. It's gonna get rid of the other. And then based on the sequence you have here, then the RISC is gonna go and find the complementary part on any gene. It's not any gene, it's a specific gene, in fact, that is having complementary part to this RNA. And then that RNA is gonna be, the translation is gonna be inhibited. And the inhibition in this process is not by degradation. The inhibition is by the fact that RISC is not gonna allow the translation process to take place because the translation initiation and propagation is not going to take place. Now, the small interfering RNA is very similar to the microRNA, but we don't know the origin of these small interfering RNAs. Some researchers think that it comes from viruses, but these are inserted in the genome. But once we have these double-stranded RNA, the dicer will recognize these double-stranded RNA as if the same situation with the microRNA here. The difference is that the microRNA has to be transcribed in order to get there, while the small interfering RNA are already like double-stranded. And it's important to remember that the dicer is really specified to identify double-stranded RNA, because normally we should not have many of these double-stranded RNAs. And one of the specificities that the dicer have is to find those double-stranded RNA. And then the process is the same. Once the dicer cuts these small interfering RNA, then one of these copies is used as a template with the RISC, and that is going to bind to a specific gene, and therefore it will degrade the messenger RNA of that specific gene. So if this is gene X and you are interested to knock it down, all you need to do is to generate a double-stranded RNA specific to that gene, in other words, complementary to that gene, and you generate it in a double-stranded fashion. You give it to the cells, and the cells will generate by the dicer. When you give it to the cells, it will be recruited to the RISC, and then the RISC is going to go to that gene using that guide. It will bind complementary to that messenger RNA, and it will destroy that RNA. And then the RNA is going to be degraded, and the gene is not going to be expressed anymore. So that's how microRNA and a small interfering RNA work. Now, for us, as I said, I just went a little bit off the road to describe to you what I have described before, and I'm coming back to the process of isolation. So this slide here was telling you that there are many types of RNA and functional aspects. They are coding or non-coding, but when we isolate the RNA, we isolate everything together. And the steps are exactly the same as we did with the DNA. Just be careful that if you're using trizol, it's going to be the acidic form. You need to do the homogenization step. So you would take either the cells or parts of the mouse or tissues, and you homogenize it. And then you will have RNA, proteins, and DNA in that tube mixed with the phenol chloroform, with the phenol, sorry. And then you will add the chloroform so that you get the separation phase, and that's why the chloroform is used and the isoamyl alcohol to give you this separation phase. And then you will do a separation of the aqueous phase, where mostly RNA is there, very little amount of proteins or DNA can be found. Take very good care if you are separating this phase. You don't go to zero, you leave a little bit here, so that you don't take anything from the interface or the lower part. And then we will add isopropanol, and that will cause the RNA to precipitate. If you centrifuge that, you will get the pellets. You get rid of that, and then you will have your RNA in the tube. So it's very easy. This takes around, if you are doing it, it takes like 30 minutes. In 30 minutes, you can do that in the lab. Your homogenization is like 1 or 2 minutes. You break it, and then centrifugation for 10 minutes. You get that phase after adding the chloroform. So here you homogenize, add a chloroform, centrifuge for 10 minutes. You have the separation. You take it into a new tube, add isopropanol, centrifuge for another 10-15 minutes. You get the pellets, you wash it, and you're done. And now you will have your RNA extracted. Be careful that the RNA has to be stored at -80. Prevent using it multiple times. If you want to use it multiple times, you have to make aliquots, so you take it and use it right away. Don't do freeze-thaw cycles. And we discussed that you can optimize the protocol. So imagine you are doing this step and you carried some here. of the organic phase, all you need to do is just repeat this and you will get a better quality RNA. Now, after getting the RNA, and we discussed that there are now very expensive kits that you can use where you can extract RNA, DNA, and proteins from the same sample. I'm not going to go to the details of this slide. I'm just reminding you that if in case you have very special and this precious samples, like mainly human samples, you can take all DNA, RNA, and proteins from the same sample, so you don't need to get rid of proteins and DNA. And nowadays, we have also kits that come to isolate only RNA, like the RNA spin columns. The process is practically the same. You will first lyse, whatever lysis method you are using, blending or homogenizer or detergent or enzymes. You combine that with proteinase K and cellulase in case you are using like bacteria or plants. And then you will get a homogenate. All you need to do is just load it on that column. Once you load it on that column and you pass it through, the DNA will bind to that column because these columns are coming with a silica that is mainly containing the guanidinium hydrochloride that allows DNA to bind specifically to the silica, and DNA, RNA, and other stuff will pass through. When you spin down, the binding takes place. And before you do the binding, you add ethanol because this will enhance the binding capability of the RNA to the silica. Then, if you want to assure that you don't have any DNA, you can do a step here where you can kill any DNA by DNases, as we mentioned before. So all you need is just add DNases onto the silica and incubate it for 10-15 minutes. And then you will do a washing step to get rid of any salts or impurities, and then you will elute with water. So washing is, again, just add the washing buffer, spin down. Elution is the same, add the water or the TA, and then spin down. So I'm pretty sure we went extensively on those, but I'm going back again and again because I'm sure if you go back to any lab and you will perform a qPCR, the first step is to isolate RNA. And if you don't take good care of those steps, you end up having RNA, but the RNA quality is bad or degraded. And it's important for you to have a good quality RNA. Let me see, do I have any questions? No. Okay. So speaking about the quality of the RNA, exactly as we did with the DNA, we would like to test the quality of the DNA by two ways. The first way is by the spectrophotometry. And in the spectrophotometry, we use the optical density to first calculate the concentration and then tell us the impurities that we might get in the RNA. So be careful if you're using the spectrophotometer to use the RNA in the settings from this drop-down menu. You know that for DNA and RNA, the optical density, the maximum optical density as 260. So this is a very good example here where you see a very pure DNA sample, RNA sample. Be careful that 260 to 280 is different between DNA and proteins, and RNA, sorry. The DNA is more than 1.8 is considered pure. For RNA, you should consider at least 2 to be pure. And in this example here, 260 to 80 is around 2.2, 0.02. Impurities that show up at 230 or at 280 are also quantified by the value of 260 to 280 value, the ratio, and the ratio should be around 2 to 2.2, which is just perfect in this example. And lastly, we can test the RNA integrity by looking at the RNA on the gel. So you can load multiple, like the gels on agarose gel, the RNA on agarose gel, and then you check the presence of the S28 and the S18. And sometimes you see the 5S as well. Now, the better the separation of these bands, it tells you that the RNA integrity is good, and that's a high RIN of 10. And that's where you can see the band here in there, and the band of the 18S in there, and the 5S somewhere like there. Now, the more degradation that start to take place in RNA, so if you load sample number 1 and sample number 12, and I think you got a question in the exam, that it's super clear that sample number 12 is degraded because, first of all, the fragments are much smaller, and that's why you see them here, not there, in terms of time. And second, you can see that the degradation of multiple bands showing up on the gel. So if you are doing a big experiment, you have to validate the RIN. And again, a higher RIN number, which is 10, the highest, compared to the lowest, which is 1, can tell you a lot about your RNA quality. Great. So I just refreshed your memory how to isolate RNA, and then what types of RNA that we are interested in to study. But now imagine we want to study that RNA, and how can we proceed doing that? So we can perform a single gene analysis level. So we can study the transcriptional profile of, when we say transcriptional profile, meaning the DNA has been transcribed into RNA. So when we say transcriptional profile, meaning you are looking at the RNA of those cells compared to RNA of other cells, like control conditions or experimental conditions, treated with a drug or not treated with a drug, from a mouse that is not sick to a mouse that is sick, from a human who has a mutation to a human who doesn't have a mutation. So you are simply taking all the RNA and you are checking to particular aspects on a specific gene, and we can do that in the lab by qPCR or quantitative real-time PCR. There are multiple types of PCRs. One of them is digital PCR that's we rarely use, or you have at least to be very good in doing qPCR in order to be able to do a digital PCR. Now, if you want to do a whole transcriptome analysis, you can do that in multiple ways. First, you can generate, for example, if you want to check the messenger RNA that is generated from 2 conditions, you can take the messenger RNAs, you can generate a cDNA library, and we discussed how we do a cDNA library before. Or you can do microarrays and RNA sequencing. And we will treat this in the next session. So for today, we're going to focus on the qPCR only. Again, for generating a cDNA library, you recall that we have to use messenger RNAs. And in order to do a conversion or reversion, if you want to say, from the RNA back to the DNA, you have to use what we call a reverse transcriptase. And these reverse transcriptases will read the message on the messenger RNA and will revert it back to DNA. And it's a very good method that is widely used to study the messenger RNA and to clone a new messenger RNA that has not been studied before. And the steps are pretty similar to what we have done with the DNA library. And we went through this many times. It's simply, the only difference is that instead of taking a DNA, we just take a messenger RNA, reverse transcribe it, and make the the double-stranded cDNA from that, and then clone it into a vector, and that vector goes to a bacteria, and we will have many, many of these bacteria. Each one is one clone, and each one has a specific messenger RNA. And with the sequencing procedure, we could study the genomics, we could study the epigenomics, and we could study the same way the transcriptomics. One way to do that is by cDNA libraries and cloning and then sequencing of the results. And the other way would be by studying particular genes or the whole transcriptome by looking at all the molecules of the RNA in the cell. So for today, we will describe what is the difference between a PCR and a qPCR or quantitative PCR. So you remember when we perform a PCR, we would need to set up a reaction which contains a DNA template, which is the DNA we want to amplify. And we need an enzyme that has to read, which is the polymerase, the Taq polymerase. We would need primers to start the polymerization step. Always start from 5' to 3' end. And we need a thermocycler because we're working in a lab, so we have to increase the temperature to 95 degrees to assure that we have denaturing. And then we do a kneading step so that the primers bind. And then after that, with the Taq polymerase and the dNTPs, the process of the polymerization takes place. And we do this many cycles, normally up to 30 cycles. It depends on the experimental setup. And this slide we have already discussed, and there's a video on that. Now, if I want to make a quick comparison between the PCR and the quantitative real-time PCR, what are the components that are common and what are the characters that we have to take a very good care when we are performing the qPCR? So again, a PCR reaction should contain the optimal buffer where the reaction is going to take place. And the optimal buffer has to have the best situation for an enzyme to be active, which is the Taq polymerase in this condition, because it's the enzyme that is polymerizing the gene amplification. We need to generate specific primers, and now you know how to generate primers based on the experimental setup, as you saw in the assignment 2. You will need to add the dNTPs so that when the Taq polymerase reads what is on the template DNA, then we will have the recruitment of the dNTPs. And then, of course, we need the DNA that we need to amplify. So if this, in the assignment number 2, you were amplifying the IGF-1 gene. So that is your template. Now, if I'm generating a PCR product for the transcript of the IGF, not for the whole gene, let's say transcript 2 or transcript 3, we will still need the enzyme and the buffer for that enzyme. And it could be different types of enzymes. Probably here it's written Taq polymerase, but different companies probably provide different polymerases. Now, the primers has to be specific for that gene, but we have to be very careful when we design those primers. I will show you in the next slide. Now, the template is the DNA could be a genomic DNA here. The template in the RNA in the qPCR should be the cDNA, which is the coding part of the gene. And obviously, when you generate a PCR here, most of the time you will take it to the gel and you check it, or you want to clone it, put it in a bacteria on a plasmid, and then do whatever. In a qPCR, you have to check how much of that DNA you identified. And for that, we need fluorescent dyes that bind to the PCR product. The more you are generating of this PCR product, the more of these dyes will bind. And then it's not a normal thermocycler, which just increase the temperature to 95, then to 55, then to 72, and then again to another cycle and another cycle. It should have these capabilities, plus it should have capability to read the amount of these fluorescent dyes that bind to the DNA. And finally, we should be able to analyze the melting curves. And we will check what are those melting curves and how we read them. So again, this slide is copied 2 times just to remind you for one last time that when we are doing qPCR, we are designing primers within the exons. So within the exon, if I have this gene in 2 different transcripts, and I want to differentiate if it's, for example, in normal cells versus cancer cells, which isoform is expressed more, I have to design primers that are specific between exon 3 and exon 1 on one side, compared to exon 1 and exon 2, or exon 2 and exon 3 on another side. Because the site where the primer binds, if it binds between 1 and 3, it will pick only this isoform, while if it binds between 1 and 2, it will pick only this isoform. So that's how you can differentiate between the two. Another aspect that we have to, so I wrote it here again for you, when performing a qPCR, it's crucial to take into consideration the transcript you are studying, i.e. the exon combination in that transcript. So when we are designing a primer for a qPCR, we have to take into consideration that we don't include any primers in the intronic region. And we don't include any of the primer in the junction between the exons and introns, or in the boundaries. Because if you look here, you have a genomic DNA. It has exon 1, intron, and exon 2. So if I'm designing a primer where the primer is binding to part of the exon and part of the intron, during the splicing, you're going to lose the intron, and this primer is not going to be efficient. Okay, so your primer has to be within exon number 1 and within exon number 2. It should not have the boundaries of intron exon at all. Because in this condition, you will not have any products. Only in condition where the primer binds to exon 1 and exon 2, that's where you get the product. So you have to make sure that your qPCR primers should not be at the boundaries, or in other words, they should be interexonic from one exon to another exon, one on exon number 1 and one exon number 2. Now, this is the first criteria. The second criteria is that the qPCR amplicon, which is the sequence you are amplifying between exon 1 and between primer 1 and primer 2, should be ideally between 75 to 300 base pairs. That's generally what we do in the lab, because if we increase more the amplicon, the efficiency of the amplification is much lower. So if you go to any paper and you read the qPCR products, you will see that the amplicons are between 75 to 300 base pairs. And again, the amplicon is the size of the fragment you are amplifying on the cDNA. So these two parameters are added on top of what we already know and we have to take care of in terms of a primer design. So we still have to make sure that the GC content, the melting temperature, and the secondary structure of these primers are okay. On top of that, they should be specific to exons, and the space between these two for an amplicon does not exceed 300 base pairs. So that's how you design a primer for a qPCR compared to general PCR. Now, there is a video here. You can watch it to have better visual memory because you have some animations there. And if you remember the gene we were working on, the example, the IGF-1 that you had to amplify in question number 9 in the assignment, I asked you, take the transcript number, I don't remember, number 1 or 2, and then amplify it. So I asked you to amplify the cDNA, which is from the first start codon till the stop codon. Now this transcript has 4 exons. They are alternating in color from black to blue. So if I'm designing primers to amplify this transcript, I have to make sure that my first primer is within the black forward, and then my reverse primer is within the blue, and the size is around 300 base pairs. So the best way to do a good PCR primer for qPCR is to take the cDNA, the coding sequence, and then keep your exon 1, exon 2, exon 3, and exon 4 separated, so you would know where each exon start and end. And then you can just take, for example, exon 1 and 2, and you can give it to the primer 3 software, and it will design primers for you. Now, say you want to design 3 primers or 4 primers for this particular gene. You can choose one primer on exon 1 forward and primer on exon 2 reverse. You can choose a primer forward on exon 2 and a primer on exon 3 as reverse. You can choose a forward primer on exon 3 and a reverse one on exon 4. So you can make combinations. Now, because I'm showing you only the cDNA, I don't have the introns here. I'm sure that I have no introns in my coding cDNA. So I have a question. I will stop to answer that. I'm not sure if this is explained in much better way than what I said here in the video. Cooper, if you didn't get it, can you specify your question so I can repeat? Cooper, are you going to specify your question? Okay. I just wanted to know about the exon 3, 4 stuff you're saying. Okay. So what I was saying is that the general concept is that if you are generating a primer, they should not be on the same exon. Why they should not be on the same exon? Because the same exon, you can detect the genomic DNA. If you put your primer forward and reverse here, this is on the genomic DNA, not on the cDNA. So this is one thing. If you put your primer between the exon and the intron, when the cDNA is generated, the primer will not bind. So there will be no problem. So the best scenario, and that scenario that we use, is one primer per exon. So if we have exon 1 and 2 here, one primer forward, one primer back. But if you have more than one exon, 1, 2, 3, 4, you can make any combination. You can use, let's say, exon number 2 as a forward. You design a primer here going forward. And then you can use exon number 3 as reverse. Or you can do exon number 3 as forward and exon number 4 as reverse. That's what I was saying. You can make any combination you want between 1, 2, 2, 3, 3, 4, or 2, 3. Sometimes if the exon is very small, imagine that the exon is only like 15 base pairs. Sometimes we have exons like that. You can't design a primer on 15 base pairs. So simply imagine you have here an exon here, only 15 base pairs. You can jump from exon in the blue all the way to exon in the black. So you can make combinations. The bottom line, I'm not going to ask you about those combinations. The bottom line for me, I want you to understand how you design the primer. So if I give you 2 exons, I want to know where you design the primer. I'm not going to give you the combinations of 7 exons. Okay. Why can't you use 2 primers on the same exon? You can't use the same exon because what's the difference between genomic DNA and coding DNA? cDNA. The cDNA comes from where? Let's go back one step. So that's the genomic DNA, and that's the messenger RNA, right? The difference between the genomic DNA and the coding messenger RNA, excuse me, it is that you lost the introns, right? So if you design your primers both in the same exon here, then you can amplify the genomic DNA as well as the cDNA. While if you design one primer here and one primer on the next exon, then there's an intron in between on the genomic DNA. And that's why I was saying it's very important, the amplicon size. So the amplicon size would be more than 300 base pairs here. So the PCR product is not going to be valid. While on the spliced messenger RNA, the exon 1 and exon 2 will be very small. There will be no spaces, sorry. Then you can amplify that, and then you can detect the messenger RNA. Now, why the amplification of more than 300 base pairs is not going to take place? Because simply we don't give the time. You know that the Taq polymerase needs some time to generate a product. For example, for 1,000 base pairs, it needs 1 minute. So we don't give enough time to do more than 300 base pairs to assure the specificity. Did I answer your question? Because this, again, Jacob, if you put a primer starting of the first ATG and ending at the end of the exon, wait, and the reverse primer, again, the simple answer for your question is that you will detect both genomic DNA and cDNA, and you don't want to detect both genomic and cDNA. You want to differentiate between that. And the only way you can differentiate between these two is by spacing them out with the introns. The introns exist on the genomic DNA. So if the primers are on the same exons, there's no way that you can space them by introns. You use, in fact, the introns that they are existing on the genomic DNA so that you get rid of any product that comes from only the genomic DNA. What were the primer parameters again for the GC? And so the GC content, 40 to 60% melting temperature between the two primers. Should not be more than 5 degrees, and there should be no hairpins. And Zekwan, if you're asking me this question now, please go back to the lectures and to the assignment. Okay, so did I answer your question? Okay, I think that makes sense. Okay, great. It makes sense that now oh, Zekwan, it's the same parameters for a good primer. However, you have to make sure where to design them. So you have to design them on exons, and you have to make sure it's not only on one exon as I was describing, so it doesn't really bind to the genomic DNA only and get you an amplification. You want to use on different exons, 300 base pair differences, and then it has to be 40 to 60 base pair, 40 to 60% GC, melting temperature difference no more than 5 degrees, and no secondary structures. So these are from the regular PCR as well. Okay. Sounds good. So now we are moving to the second step. So we designed primers for the qPCR. And if you use primer 3, it gives you suggestions. All you need to do is just to take those and to test them. Now, the second modification from a regular PCR to a qPCR is the usage of the fluorescent dyes. And there are two types of fluorescent dyes that we use. The fact that these fluorescent dye would bind to the double-stranded DNA, not to the single-stranded DNA. So, for example, Cyber Green, which is the cheapest, and you can find it in every lab, it binds only to the double-stranded DNA. But the binding, be careful for this particular information. The binding of the Cyber to the double-stranded DNA happens while we are generating the PCR. Because recall, when you are doing a PCR, where is it? Here, you have a double-stranded DNA. So if you take this double-stranded DNA and you just add the Cyber, it's not going to bind to it. While if you do the denaturation, so it becomes single-stranded, and while you are generating the double-stranded, then the Cyber can bind. Okay. So it requires thermal cycles, as it says, that incorporate the ability to detect the accumulation of the DNA in a real-time fashion. So each time we have a PCR product or one copy that is copied, you will have more of those dyes bind to the DNA, and therefore you can see that there is more fluorescence being generated. So if you have one copy, you have only one copy of this, and imagine in the next you have two copies, then the fluorescence increase, and then the third copy, more of the fluorescence comes. And as we get more and more accumulation, then you will have more and more signal, and then you can compare between two different conditions. There are very good 2 YouTube videos for you here. You can see how the process of qPCR accumulation takes place with the Cyber Green. Now, the Cyber Green is not the only way we can detect a PCR that is being generated. So here, it's very cheap. It's efficient, but it's not specific. Any secondary, any double-stranded DNA that is being transcribed, then we will get a product of the fluorescence that is being added. So scientists find a different way to get more specific, and this would be by using of the Tachman probes. I'm sorry. And the Tachman probes, we will have the capability to have the probe, which is a specific sequence that binds to a specific gene. So when you say a probe, as we defined it before for the southern blotting, it's a specific DNA sequence that binds to a specific gene. But on the extremities of this probe, you added a reporter and a quencher. And since the reporter and the quencher on that probe are very close, there is no fluorescence to start with. Now, then you designed primers that are specific for that gene, and then you start the process of the amplification. When the polymerases start polymerizing the strands, it will reach somehow to the probe, where the probe is bound to the DNA. Obviously, I have to tell you that the probe will bind only when you do the process of the denaturation. So you have to denature the DNA, so all the DNA is single-stranded, and then you will do the annealing step, and during the annealing step, the forward primer, the probe, and the reverse primer all will bind to that DNA. And then the polymerization start to take place. When the polymerization reach to the site of the reporter, is gonna cleave it. And that would lead to the release of the reporter. And of course, it has exonuclease activity, so it will remove this probe. And after that, this reporter is away from the quencher, so the fluorescence become active. So the more you do of this PCR product, the more you get of this fluorescence. So the only difference between the Cyber and the TaqMan is that the TaqMan is already binding to a specific part of the gene, so it's very, very specific, the sequence. While the Cyber, it binds to any double-stranded DNA you are generating. Now, the more of the fluorescence you get, the more that the machine is going to record that you get more fluorescence being active. In other words, the more amplification of your gene that is specific. Okay. Any questions here? No questions so far. Okay. So then, how do we read the results of a qPCR? So if you set up a reaction, you put all the components, which including the NTPs, polymerase, the primers, whether you are using Cyber or you are using TaqMan with the primers, and you start doing the cycling, which is denaturing, reannealing, and amplification again and again and again. And you start reading 0 cycles, 1, 2, 10 cycles, 20 cycles, 30 cycles, and 40 cycles. Generally, there are 3, sometimes like people merge 2 of the phases into 1. Sometimes they say 3 phases or 4 phases. I just present for you the 4 phases, which is the first phase. You don't see any amplification because you don't have yet a threshold of detection of the fluorescence. And then the start of the amplification, which starts with the second phase, but yet you are not in a linear setup or not in the exponential phase. And then the third phase, where you have the exponential amplification of your product. And finally, you get to a plateau where there is no more amplification. And generally speaking, a real-time PCR normally runs to a maximum of 40 cycles. Generally accepted anything below 30 for the protein coding genes. And this is a typical qPCR curve, where there is no PCR product detected, and as you add more and more cycles, you start to have amplification. The more and more cycles, this means you are adding more and more fluorescence to the double-stranded DNA, or in condition of the TaqMan, the reporter has been released and you get more fluorescence there. Now, generally, when we are identifying a qPCR product, we would like to study the exponential phase. And the number of cycles are very important to describe how we get to the product and how we can analyze the products. So far, there is nothing really crazy about what we added in terms of the qPCR compared to the general or regular PCR. It's simply just the fluorescence that we are detecting here and some specificity of how you design the primer. Now, the first thing you have to do is to check if your primer is specific. And in order to do that, you can do what we call a serial dilution of the template cDNA. So simply you take the cDNA and you make 1:2 dilution, or you diluted 1/2, you diluted 1:4, you diluted 1:8, and you diluted 1:16. And then you perform reactions in triplicate from this, in triplicate from that, in triplicate for 1/8, in triplicate for 1/16. There are many other triplicates here, but I'm just giving you an example. Then obviously the more concentrated DNA will generate a product much faster. So if this is non-diluted DNA, this is the 1:2 diluted, and this is 1:4 diluted, and this is 1:8 diluted, and this is 1:16 diluted. So the more diluted the start material, the longer time it needs to generate the PCR, and the longer time you detect the PCR product. And here we are just looking at the exponential side of the, we are just looking at the exponential part of the curve, which is from here to there. See, we of course we can see the plateau, but I mean, you don't see the lower part, the non-expressed. So replicates of the same reaction are analyzed, because the replicates can tell you if you make any errors by pipetting. For example, you put more or less volume. The CT values of replicate indicate low technical errors, such as pipetting errors. Well, I wrote it down. And then if you blot the threshold of cycles, you can get, in terms of how many cycles you need to generate how much of the DNA. In other words, the more cycles you need in order to amplify a DNA of the lower concentration. So if the concentration was 2, let's say, femtograms of DNA, you would need at least 30 cycles to amplify it. If you start with 3 nanograms or 3 femtograms, you will have 25 cycles to amplify that. If you start with 4, you would need less. If you start with 5, you would need much less, and on and on. So when you generate this curve with different templates, the same template, you are doing many dilutions with the same set of primers, then you will have this separation per cycles, and then you can generate this curve because you will have the number of cycles and the concentration. And you know this mathematically, when you generate a curve, you can get an equation where you know the slope and you know the R square, and this can help you to establish the concentration of the cDNA you started with. Okay. Now, how does it look like when we test the primer specificity? The last step of a qPCR is to do a melting, where you will increase the temperature from 55. So this would be the last cycle. The machine is gonna stop there and it starts raising the temperature more, 50, 60, 70, 80, and suddenly you will get this format of the curve. Now, so why we get such a curve here, and what does it mean? So as you can see, I wrote it down. The change in the fluorescence with the increasing temperature is measured. And that's the last step in the qPCR. As the temperature increases, the 2 strands of the amplicon, which are now amplified and they have the fluorescence dye, they start to tend to generate a single-stranded DNA, because you are increasing the temperature till the point where you will have your amplicon, which is a double-stranded with the dye is gonna now reach its melting temperature. When it reaches the melting temperature, it will go to a single strand. And once it gets to a single strand, it's gonna lose directly all the dye, because you remember the cyber is not gonna bind to the single-stranded DNA. And now, if you get this type of curve in your primers, this means you are amplifying only one product. And this means your primers are very good to use. However, if you get this type of melting curve, it will tell you that you have two different products. One that melted around 75 and another that melted around 85. And therefore, this primer you cannot use in a qPCR experiment. It's not specific to the gene you are using. That's how the primer should be in order to use for a specific gene. Okay? How many minutes I have? I still have few minutes. I will go quickly on a couple of information, and then I will refresh your memory again next session. So in other words, what I showed you here, it tells you that any product you get in the earlier cycles is more concentrated or more abundant compared to something that show in the higher cycles. So in other words, if you have a condition where a gene that is normally expressed at this level in wild-type cells, and it's lost with the cancer, like p53, we many times discussed the p53, the tumor suppressor gene, is lost in cancer. So if you compare normal to cancer, the normal cells will have the expression start around here. And in cancer cells, since it's deleted, there will be no messenger RNA and there will be no cDNA or much less, and therefore it needs longer time to show up. So that's how we use the qPCR to differentiate between a tumor cell versus a wild-type cell. Now, these three conditions, again, the high expression identified in lower cycles. So this means in the lower CT, the cycle threshold, for this particular gene X is expressed the highest in the condition number 1 because it showed up much higher compared to conditions 2 and 3. Now, you can imagine that, for example, someone who is normal for insulin production has this curve for insulin gene. Someone who is pre-diabetic, so is still producing some insulin, but not much, it shows up here. And someone who is really diabetic, who lost the expression of insulin gene, he needs much longer time to show up for the expression. So that's how we use qPCR to profile the transcriptional aspects of a disease. And again, this is quite correlated to the quantity of the cDNA. In other words, you would need more cycles. Number 2 here is most probably correlated with the green because it's much lower cDNA. You need much more cycles to amplify. And then probably here, number 4 is correlated with the yellow. You need slightly less than the green to be amplified. And the high concentration, or in other words, the high expression is correlated with the red. Okay. Now, if you are taking the example of a normal cell to a tumor cell, like for example, if we're looking at the red transcript and we're having a primer that is specific to the red transcript, ideally, you will have much more in a normal person compared to someone who has a tumor in terms of the red transcript, because from the tumor cells, you have only one copy here, and you need much longer time for that cDNA to amplify and generate the cycles, while in a normal cell, you have many copies to start with, So it will be much faster to generate that product. Okay. So I think that gives you a good indication how we can use the qPCR. Now, in order to make a quantification, we can do two different types of quantifications. We can do the absolute quantification, which would require this curve. So we can say that in this condition, when we get the curve, imagine you want to know the expression in one of the conditions, but you want to know exactly how much amount of DNA that you started with. So to do that, we need to generate this curve, and we need to get the slope and the R squared. This is statistics. You should know how to do that. And simply, if you have an unknown that comes in cycle, let's say 22, all you need to do is just to project and calculate how much DNA you get. And that would be an absolute quantification. So the first way is the absolute quantification, which requires the calibration curve that we discussed. The second way is the relative expression. And in that situation, you will take a cell and to compare it to another cell. And you will say, this gene is expressed 3 times higher compared to that in the tumor cell. So the red gene is expressed 3 times more in the normal cell compared to the tumor cell, or other way around, the red gene is 3-fold less compared to the normal cells. Okay, and the way we do these comparisons is by having also housekeeping genes. So what does it mean, a housekeeping gene? These housekeeping genes are used as references. Sometimes they called reference gene, sometimes called control gene. Some of those that we widely use in labs are S16 and GAPTH. So these are enzymes that are used in cellular maintenance and are essential for regular, basic, and ubiquitous cellular functions. And normally they should not change from a normal cell to a tumor cell. So we need to have those that don't change between the two as a control. And I will show you in the next slide how we use them. But before moving forward, I want to make sure that you guys know that many of these genes people use, and they think that GAPTH, for example, doesn't change between normal cell and tumor cell. Or S16 should not be different between these two. And we use it as a control. But unfortunately, with research, we find out, no, it's not the case. So you have to be very careful which gene you are using as a control. And if you are doing a qPCR and your PCR product is all over the place, think about the control gene you are using. Okay? So how do we quantify in terms of the absolute numbers? The absolute numbers I just described, you need to perform the qPCR, you need to generate this curve, and from this curve, you will get many standards, and all you need to do is just to say, oh, this sample is, for example, on cycle, let's say, 22, and from there, we can calculate. We can know how many cycles we have, and we can calculate based on the curve. If it's cycle 22, then we had 5 nanograms, 5 femtograms of DNA. So that's an absolute way. We don't use this too much in the lab, to be honest. And the most widely used is the Amplicon relative analysis. Now, in the relative analysis, though, bear with me 1 minute how we do the analysis. So I was telling you about the housekeeping gene that should not be different between the experimental conditions. So we have 2 conditions here. Let's say a control group, cells that are not treated with anything, HeLa cells or HEK cells, whatever. and cells that you treated with a drug, whatever drug that was. So you have a control condition and experimental group. So in principle, the reference gene or the housekeeping gene or the control gene, they should not be different between the conditions, the control condition and the experimental condition. So S16 and GAPDH should not change, and that's why they came together at the same time. Okay? Now, the experimental condition, it seems to be amplified faster compared to, the experimental condition seems to be amplified faster because it showed up on cycle 20 or such, while the control condition, it needed longer time. It showed up on cycle 20 something. Okay, so I can directly say that, oh, I have very good control genes in both conditions that didn't change, which is S16 or GAPDH. However, my experimental condition seems to be overexpressed or upregulated compared to the control condition. And the way we do that, we get the delta CT threshold. So we get the comparison of the experimental condition to the control gene in the experimental condition. This is number 2. And then we do the cycle differences between the control condition and the reference gene. And that's going to be number 1. So you get the delta CT. So simply the difference between the cycles, those appeared. And then you get the delta CT, which is the difference between the first group to the second group. And that's exactly what people use every day in a qPCR analysis. I know the last part was quick for you. I will repeat this in the next session. And if you have any questions you want to stop for now, we can stop and answer those questions.
Professor.
Just give me 1 second. I would like to stop the recording.